Myself Divyasree, I'm gonna tell about the physics of enzymes, and my topic is about the chemical aspects of enzyme activity. Coming to the main point, what is this enzymes means? Enzymes are biological catalysts that speed up chemical reactions in the body without being consumed in the process. The action of enzymes involves several key chemical aspects like active site, substrate binding, transition state, product formation, cofactors and coenzymes, and enzyme regulation. The main characteristic feature of enzymatic catalysts are the high activity and strict specificity with respect to the substrate or a group of substrate. The molecular activity or turnover number of enzyme is defined as the number of moles of substrate converted per minute by moles of enzymes at a considerable excess of substrate. Mainly substrates are small molecules or small groups of large molecules. On the contrary, enzymes are macromolecular substances, hence the substrate interacts directly with a definite small portion of the enzymes called an active site. The nature of the active site, that is amino acid composition and the sequence and also the cofactors that enters its composition, has been established for a number of. Coming to this, we are able to see a diagram which is related to a systematic illustration of the action of creatine and kinase transition states. From this figure shows the schematic of transition state for this reaction. The cofactor is the magnesium ion. An especially important role is played at the AS and AS by the SH group. The enzyme is inactivated by iodoacetamide and iodoacetate in the presence of the magnesium ion and both substrates. The SH group is protected from the action of blocking agent. The SH group is attached through a hydrogen bond to one of the main groups of the enzyme. The factal transition of a proton in the hydrogen bonded acid-base system imparts high catalytic activity to the system. The electron transfer in substrates and groups of the active sites are shown in substrates and groups of the active. Start. The active site is naturally chiral and thereby stereospecific. It has been found with the aid of labeled atoms that reactions of molecules of the type, that is CABD, occurs on the surface of the enzyme in an asymmetric manner. This refers for an example to the transformation of amino malonic acid to glycine. See, this is the figure of this amino malonic glycine, and this will convert it to this, and the here the carbon which is represented with the star, it is labeled as the carbon atom, and the reaction involves only one COOH group, that is carboxylic group, which is chemically and geometrically indistinguishable from the other group, and this is accounted for by the asymmetry of the active site. Next, the direct X-ray diffraction study of complexes of this lysosome with inhibiting analogs of substrate polysaccharides has shown that the ligand is incorporated with an cavity present in the lysosome globule and comes into a contact with the several functional groups of the enzyme. So now next, coming to the reaction steps taking place at the active site of the enzyme substrate complex. For instance, Bronstein and his colleagues have investigated the chemistry of aspartate aminotransferase. This enzyme contains pyroxyl phosphate as a cofactor. Pyroxyl phosphate added to a protein reacts with the substrate and amino acid chemically forming, that is algimine. This is called as the Schiff base. Coming to this, we can observe here the hydrogen alpha atom dissociates readily in almidamine in transamination reaction, aldehyde lactomerase to ketimine. See, this which is hydrolyzed by water to give the ketoic acid. Now next, coming to the topic, that is there are some qualitative factors which was proposed by the Bronstein, and he pointed that like there are some of the 5 rules of this. Coming to the first one, a high affinity between enzyme and the substrate, that is a high probability of formation of an enzyme substrate complex which is equivalent to a sharp and increase in concentration of reactants, that is called the proximity of effect. A strict mutual orientation of reactants, cofactors and the active site, the orientation, this is called the orientation effect. In ordinary homogeneous reactions, the probability of 3 or more interacting molecules being strictly oriented with respect to one another, this is very low here. The action on the substrate of nucleophile and electrophile groups of the active site, that is the effect of concerned general acid base catalysis. Coming to this figure, we can tell that the hydroxypyridine catalyzes the mutarotation of glucose, rupturing its 6 membrane ring and undergoing a tautomeric change. See here we can observe this tautomeric change which is undergoing, and this is the analogs reactions involving a mixture of pyridine and the phenol, and proceeds at a rate of 7,000 times slower. The cooperation of an acidic and the basic groups, their concerned action sharply accelerates the reaction. The activation of the substrate through the redistribution of electron density under the influence of active groups of the enzyme, that is called the polarization effect. See the last point, that is called the induced Fichte effect. What is this means? The change of the conformations of the protein and substrate in the enzyme substrate complex. This is called the induced Fichte effect. Next topic is about the conformational properties of enzyme. In this, the physics must be concerned with the quantitative aspects of these pictures, indicating at the same time to what extent the effects resisted above can be represented. See, there are some of the enzyme non-enzymatic counterpart, rate of enzymatic process, rate of non-enzymic process. See, coming to this, if you take an example of the lysozyme, its non-enzymatic counterpart, it is a hydrolysis of acetyl base catalyst, and rate of enzymatic process, that is 5 into 10 to the power of minus 1, and rate of non-enzymatic process, that is 3 into 10 to the power of minus 9, and that is V by V naught value of this is 2 into 10 to the power of 8. So like this only we can find out all these non-enzymatic counterparts and rate of reactions of all these, like for beta amylase and fumarase, all this also. So next, there is an M.L. Fisher. He is one of the scientists. He proposed the lock and key model for the treatment of the enzyme substrate interaction, and the enzyme and the substrate possesses rigid structure, and the active site of the enzyme being structurally complementary to the substrate, so that it fits with the substrate in much of the same manner as a key fits into a lock. See, Koshland advanced a induced Fichte theory to account for the specificity of the enzyme substrate interaction. Here we can see some of the diagrams. In these diagrams, this part is called the enzyme, and this is a substrate here which is coming inside it, and in the B figure we can observe that enzyme substrate complex is there, and C and D figures tells us about the complex with substrate analogs. These are the two complex with substrate analogs, and some of the basic postulates of this theory, which was designed primarily in an attempt to explain the specificity of enzymes that catalyzes the bond transfer reaction. See, B minus X plus Y, this gives rise to B minus Y plus X. So this is the basic reaction which takes place here. The substrate causes changes in the geometry of the enzyme as it fits into the active site, and a delicate orientation of the catalytic groups is required for enzyme reaction. The substrate induces this proper orientation by the change it causes in the geometry of the enzyme. According to the Koshland model theory, to the substrate specificity and catalytic efficiency of the enzyme, these are closely related, but their mechanisms are different, and the reaction can be accomplished only with a definite decomposition of the contact and the catalytic groups of the enzyme with respect to the substrate molecule. For example, here we can observe this figure is related to the beta amylase, and this is a non-reactive complex, and these are called the cycloamylose, and this is the enzyme part which is interacting with these part of this starch, and this is called the reactive complex which is interacting with this enzyme. The next is the X-ray diffraction studies of this enzyme substrate complex yield direct information about conformational transformations upon incorporation of the substrate into the lysosome. The Krebs contract of squeeze the substrate more tightly. The displacements of the amino acid resides of the protein, these are not great, but they are appreciable. The resides there is shifted by 0.75 nanometers. This is accompanied by a change in conformation of the substrate analog, a slight rotation of the carbohydrate rings about the glycosidic linkage, and in this we can observe that the active site of the carboxypeptidase and the presence of a Krebs design specifically for this substrate has also been detected for other enzymes, and this reveals a group of the active site. A direct confirmation of the induced Fichte concept has been obtained in an investigation of the carboxypeptidase. Thank you, ma'am.